So, this is a presentation on functional studies to investigate cell behavior in cancer. So, to start with, what actually are functional studies? Well, functional studies refer to a variety of techniques used to gather information about what might be the cause or the function of something. So let's say we're interested on the impact of a particular gene. We can do various things to upregulate it, downregulate it, knock it out, overexpress it, everything like that, and then we can actually observe these using functional studies. So why is it important to be able to study cells in regards to cancer? Well, of course, ultimately we're able to better our understanding of cancer biology, which in turn helps us with drug development. So what are the hallmarks of cancer? So I've put some up on the screen for us to see there, and knowing that these are the hallmarks, we can actually observe these using various assays. And I'm going to give us some examples focusing on migration and invasion. So what is migration? Well, it's basically when a cell moves from one area to another to exert its function, which is based on actin polymerization, where we have our lamellipodia with its filopodia protrusions. It crawls along the substratum looking for integrins to drag the cell across, and that's basically what migration is. So how is this related to cancer? Well, of course, as cancer progresses, the cancer cells are able to acquire a more aggressive phenotype, and they're ultimately able to invade and undergo epithelial to mesenchymal transition, where they can now do metastasis, and cancer cells can move to distant sites of the body. So we can use a few different assays to actually observe this, to observe migration and invasion. So firstly, looking at the in vitro 2D cultures, we can actually perform what is known as a scratch assay, which is very, very simple to do. You can observe it using real-time microscopy, and it's cheap because you just need to plate your cells on a dish, and then you can either use a pipette tip to scratch the surface, which you can see in figure 3 option B, or you can be slightly more accurate and use a silicone insert. And so the results that you'll see is, in figure 5A, we have our cell-free zone. In B, we can see that cell-free zone has decreased in size. And by C, we've had full proliferation and migration of the cells to close the wound, which you can see here. We can also use a transwell assay, which is a way of looking at lateral migration, whereas the scratch assay was our that was our lateral migration, but we can use transwell assays to look at our vertical migration. So here we have our transwell assay set up. We will seed our cells on top of the permeable membrane, so there will be lots of pores in it, and in the lower well, where we will have our test compound, whatever that may be. And if the cells are able to migrate through the pores of the membrane, then you can stain them with something like cresol violet, which we can see in figure 7, and you can count them using a hemocytometer. And you can see here that these cells have in fact moved and migrated towards the chemoattractant. But we can make slight modifications to this assay where we can observe invasion, where now in between the membrane and the cells, we can add basically something that's supposed to mimic the extracellular matrix, such as adding matrigel on top of the membrane. So the cells have to now, if they're able to invade, they have to now degrade what is essentially the extracellular matrix to be able to pass through into the bottom chamber. And so we can see in figure 8 that this was a set of results where they were looking at prostate cancer cells, and they've transfected with a particular gene of interest. They were interested in this gene known as tissue factor pathway inhibitor 2, and they wanted to see what it did in regards to invasion. So that's what we can see in S1 and S2 of figure 8. They've transfected tissue factor pathway inhibitor 2, and the invasion has significantly reduced. This was then explored even further recently using breast cancer cells, where again they've transfected tissue factor pathway inhibitor 2, and you can see that the scratch assay, the wounding area has not closed as great as in the control, so migration is reduced, and the same was seen with invasion, that was also reduced. We can also look at other things, so other assays, for example, this is to observe tumorigenesis. We can use a clonogenic or a colony formation assay. Here they've used a particular technique to silence a gene of interest. So they've used siRNA against YAP2 and YAP4, and they've noticed that the colonies in both of these cell lines have reduced. We can also look at cell proliferation. So there are multiple ways to do this. We could use a cell counting kit 8. Again, we're using the same silencing technique, and we can see that the proliferation has decreased in both of these graphs. We can also look at Pcna staining, which is our proliferative cellular nuclear antigen. Here we wanted to observe the effect of particular compounds, so they've used peptide 17, and they found that Pcna staining decreased, so proliferation decreased. Another method of looking at proliferation is by staining for the proliferative antigen Ki67. Here we can see in the control nothing. When we've treated with FGF2, there's an increase, a quite obvious increase in staining. And when we've treated with the inhibitor PD173074, there's nothing. And when we've treated with FGF2 and the inhibitor, it's basically the same as just the control or just the inhibitor. So we can say that FGF2 does in fact stimulate proliferation, whereas the inhibitor does inhibit it. We're also able to look at apoptosis. So here this is a tunnel assay, and you see on the right, when they've used peptide 17 again, it has increased the number of tunnel positive cells, so it's increased apoptosis. Whoops. So that was just an example of 2D cultures. There are they're very, very convenient and they're very cheap to do, but however, they're not very representative of the tumor microenvironment. They're not representative of the in vivo situation. And that's what led to the development of 3D systems. So basically, there are several limitations to 2D cultures. So because it doesn't mimic the in vivo environment, this is for a lot of reasons. Firstly, in 2D systems, we're using a monolayer of a single cell type. That's not what happens when we have cancer and tumors are formed. Tumors consist of multiple different cell types, and there are multiple layers. And because it's a monolayer, there's no cell-to-cell interactions with various cells that would be found in a tumor. And these cell-to-cell interactions is what drives proliferation and gene expression and responses to drugs. It's also going to cause the loss of the diverse phenotype that would be seen in a tumor. So really, it's not very representative of what would be happening in the body. And that's why there has been significant advances in the production of 3D systems. So I've got some examples here which are supposed to build on that ability to be more representative of the in vivo situation. So we have organotypic cultures, which we can see in the right. This is a cervical organotypic, and these are basically organs in a dish. So they contain multiple different cell types, and they are scaffolded onto a scaffold, so it allows for those cell-to-cell interactions. So it gives us something that's more like it gives us a morphology and architecture that's more similar to what would be happening in vivo, as well as giving us those cell-to-cell interactions. Then we have spheroids, which are have given us an organoid on the screen, but spheroids are similar to organoids in that they form 3D aggregates, so they can be made from one or multiple cells. So these are ideal for investigating drug candidates due to their ability to form these aggregates, which are more like the tumor, which represent the tumor morphology more accurately. And then we also have organoids. So like I said, similar to the spheroids, except these are made from stem cells. So naturally, they are now able to self-renew and form more complex structures. And they're able to self-organize as well. And this figure 16 is showing us it's an image that was taken from a 2018 paper where they were able to produce 95 patient breast cancer cell lines from 155 primary breast tumors. And what they found was that these organoids that they actually produced retained a number of histopathological features that were found within the parental tumors. So this is really, if that means anything to you, hopefully you'll see that this is imperative in being able to make advances in personalized medicine in terms of various things, and in this case cancer treatment. The problem with this is that these are much more complex to conduct compared to 2D systems, and they're also very expensive. So in conclusion, I've spoken about how we can manipulate genes, and we can do this through various means, and this allows us to see what effect it has within a cell, a tissue, or an organ, and we can observe those changes using functional assays. I've talked a bit about 2D cultures and how the production of 3D cultures is useful because they're more representative of the in vivo situation. With that being said, 2D cultures are very useful because they give us some sort of preclinical evidence to allow us to move on into our 3D cultures, and from there we can move on into animals. The problem with this is that you, as scientists, you want to use as little amount of animals as possible, which is why we're continuing to make advances in the production of cultures. So we have now these things that are built on from 3D cultured organoids known as microphysiological systems. And essentially, these are adding in another feature which isn't seen in organoids, and that is microfluidic circulation. So this is allowing us to have an even more complex, more representative in vivo environment. So the whole function of these is to basically have organs exist on chips. And ultimately, at one point, we're wanting to be able to make on a larger scale full humans on a chip, where you can basically have all of the organs on a single chip, and we can then observe things like metastases, for example, where there has been a study that I read where a breast cancer cell was able to we were able to observe interactions with the liver. So great advances have been made, and of course these are all invaluably helpful when it comes to anti-cancer drug development.